So welcome to the Southern Pulse Agronomy site in the Wimmera this year. We've shifted our trial site for many growers that have followed us over many years. We move around our site, Wimmera, from out the Rapanyup district, Minyip, Horsham. This year we're out in the West Wimmera, so we're out here at Nhill, and something a little bit different. This trial site was more set up to compare a couple of different soil types, and so we've got we're standing up here on a sandy rise, and we're comparing that with a black traditional clay in the Wimmera down on the flats further over. So this site has the usual array of Southern Pulse Agronomy trials, and so that'll include germplasm comparisons, disease management, a little bit of herbicide work in there, and just looking at the growth of a range of pulses. So as we walk around this site, I hope you'll enjoy the quick tour that we've got. One thing I do want to do up front is just thank the grower, thank Alan Bennett for his willingness to partner with us, and also for the funding bodies, for GRDC, Ag Victoria. Just incredibly grateful for the support that we've received over many years, and the growers that continue to back us with the trials and that we put together. So thank you very much for that, and on with the tour. So I'm just out here standing in the farmer's favour bean crop, which is potting up quite nicely at this point of time. As you can see, we've got plenty of height in it this year, believe it or not. Yes, I am standing up in here. So a fantastic favour bean crop sitting right next to our trial site here at Nhill. So something pretty unique for this trial site. We've got a sand. So for those in the Wimmera, something that we haven't done much up before, we're just working up on this sand hill. It's a key limitation for pulse production in the Wimmera here. So the grower, Alan Bennett, he generally only grow RM4 vetch up on this hill. He has actually tried growing beans before, but they tend to fail. So what we wanted to do this year is explore a number of options on this hill, and that includes ripping, and also looking at the different varieties. So I'm actually standing here right at this point in a trial where we're looking at ripped versus unripped, and vetch, and a number of different crops. This particular trial here is replicated at a number of different sites, so we're just looking at biomass development of vetch particularly throughout the season, in comparison to beans, peas and lentils and lupins on this particular sand. But the unique thing about this site is that we also have a ripped treatment. At this point of time, the ripped versus unripped, there's not a whole heap of difference. So we ripped this soil to about a depth of 40 centimetres to try and avoid ripping up any clay, particularly as we headed down towards the flats in the distance there. And as the season progresses, we expect to see greater differences as everything starts drying out. So looking forward to seeing what the results are. Not much we can say at this point of time, but certainly look out for the results come the end of the season. We're standing here in a vetch inoculation trial. So as part of the Southern Pulse Agronomy program, you'll have noted that we've got a whole heap of vetch trials in this year, from germplasm, disease management, sowing issues, so sowing time, sowing rate, plant densities, and such like. And this particular trial here is obviously looking at inoculation, and in particular what we're really interested in this one is seed dressings and impact on nodulation. So this is in partnership with Adelaide Uni and SARDI in South Australia, and just continuing to explore and maximise the profitability of vetch through maximising nodulation and inoculation. We also have another trial just further down the hill here, looking at lentils, and up further on the hill looking at chickpeas. So again, as we're highlighting before, it's about the whole package and putting the whole package together. So one of the things we just wanted to highlight here with this sort of image is some of the nodules that are going on in the vetch. You can see the massive plants, relatively small root systems, but plenty of nodules going on down there, hopefully fixing lots of N. And as we indicated, this is probably the best crop on this particular sand, which is slightly acidic, but a very deep sand for this particular region. Many other crops really struggle. So up on the hill, the chickpeas are really struggling, but the vetch is going really well around this place here. So we're just standing here in the lentil inoculation trial, and while they've nodulated quite well, we had some significant rainfall events recently, and we've just come down from on top of the hill up there and down into this flat, and you can see how it's got really wet even on the sand, and it almost looks like the lentils have been slightly waterlogged, and the roots tend to match up with that in terms of they've gone incredibly black. And if I break open these nodules, they don't look very alive. They're struggling immensely, and so what we have is a very sickly looking, poor looking lentils growing down on this sort of area here. So even though it's the sand, should be free draining, the water's come down off the hill, looking very wet down here, lentils getting potentially waterlogged. So as part of the Southern Pulse Agronomy site this year, we've got a whole heap of germplasm trials which compare the growth of a whole range of varieties and new breeding lines on this sandy soil that we've been talking about earlier, in comparison with a clay soil, which we'll be at in a few moments time. And so we're just standing here in the beans and looking at a whole range of different varieties and just looking for that opportunity to maximize production on these sandy soils, because obviously growers don't want to be constantly switching crops and having to sow a vetch up on your sand risers and then go and put your beans out on your flats over here. Ideally, if we can develop a variety, combine it with some improved nutrition, maybe some ripping and inoculation and other treatments, then potentially we can get the crop across the whole paddock and be confident that at least we can produce something on these tougher soil types. So we're standing up here in the lentil germplasm trial. So this is up on the sandhill. You'll see how much better the lentils are on the flat in a few moments time. But I just wanted to stand here because you can see massive differences between different varieties. Now I'm not going to highlight which varieties which at this point of time. Don't want to incriminate any germplasm until we get to the end of the season. But you can see the really poor growth down here in the plot. So some plants are barely surviving. Next to it, we've got another plot where things are growing quite well. And then you've got the variation as we go around. So plants just over here, which still are struggling a little bit, but going a lot better than just behind us there. And back to another good plot over this side here. The other thing I wanted to highlight in this trial as you scan around, everyone will be going, oh, look at the weeds. Yep, that's the big problem with growing lentils up on some of these sands and the competition. And also that early vigor. This trial really struggled early on in the sand, really struggled to get going. And hence we have got a whole range of weeds up here. Vetch is probably our worst enemy. But as we highlighted at the start, vetch is the thing that grows really well up on this sand. So certainly some opportunities and challenges going forward. Hopefully we get the opportunity to further explore some research solutions to growing lentils across the tougher soil types in this region in the future. So here we are down on the flat of this Nhill trial site. So as I dig down here, you'll see this is more the traditional Wimmera black cracking clay, much harder to dig into. Bit of yellow stuff down on the bottom there. Even got a nice worm crawling there for us. So a lot heavier soil, and there's been a bit of waterlogging down here just in the last week or so after those heavy rainfall events. So as you look at the overhead shots of this particular site, you'll see little water patches. Lentils have actually started dying out in certain areas. So it's quite an interesting site in terms of that.
Hi, I'm Josh Fanning, Pulse Pathologist at Agriculture Victoria. We're today at the Nhill trial site, or the Nhill Southern Pulse Agronomy site, and on this trial site we've actually got 6 trials related to disease management. So these are funded within both the pathology program at Horsham as well as the Southern Pulse Agronomy trial. We're in the middle of a vetch disease management trial, and this trial here is looking at several varieties, and we're just comparing a complete fungicide strategy with a untreated or no fungicide treatment strategy. So right here we've got a large amount of botrytis, and we're just trying to assess the yield losses due to vetch diseases. So early on in the season we were getting ascochyta blight, which we did some assessments, and we're getting 30, 40, 50% leaf area affected due to ascochyta blight, and the vetch just grew out of that disease. And then now we're starting to get to the botrytis end, and we're just trying to assess, are there yield losses due to diseases in vetch? We all hear it from growers, but we need trial data and varietal information to be able to prove what the yield losses are in terms of a percentage, so we can move forward with our disease management programs and justify them. So off to my right we have a bean disease management trial. There's not much going on here in Nhill with beans compared to say Jimbown or Lake Lithgow, but really what we've got going on is still the same trials. So if you're interested in more on those bean disease managements, have a look at Jimbown video and we'll be able to go through that in more detail. Behind the bean disease management trial, we've got some soil-borne disease trials. So we're looking at a range of seed treatments or in furrows in beans and chickpeas here at Nhill to work out if we can mitigate the effects of soil-borne diseases or see some yield increases due to these chemical treatments. Over the back here, we've got a chickpea disease management trial. This chickpea disease management trial is very similar to the one at Horsham and Currio. The Horsham and Currio ones are in the 3rd year, and this one's slightly different because we've added on trichoderma, so a biological, to see if we can control chickpea ascochyta blight with trichoderma. And we've also included a new Syngenta product in there, and we're testing whether spraying that new Syngenta product post-infection after we've seen the first signs of chickpea ascochyta compared to a strategic approach before we see the signs of disease, and comparing that with some of our other chemistries. The other trial we've got here at the Nhill trial site is a pea bacterial blight study, and so we've put out a range of treatments that we think are predisposing factors to bacterial blight in field peas. So we've got a range of chemical residues, and we've got a couple of different varieties, and we're trying to see the effects of some of these chemicals or residues on the establishment of bacterial blight in peas. Unfortunately for disease management, this trial hasn't resulted in any bacterial blight, but that's also a good thing for growers because growers in the local area haven't been seeing bacterial blight either. But hopefully in future years we can investigate bacterial blight in a better fashion or with more disease in a more conducive year.
So we're standing in here in a trial which looks at nodulation. This trial is actually supported through SARDI. They conduct all assessments out here in partnership with ourselves. But what we're looking at is the impact of certain herbicides on nodulation and end fixation. We've got all 4 crops out here. So we've got chickpeas there, faba beans in the background here, lentils 1 step over, and then vetch further back. And so I'll get Liz from SARDI to provide a bit of a background to this trial. You'll also see some canola in there, that's so we can get some infixation numbers. But you can see here some healthy nodules on the beans as we come in closer. So some healthy nodules just around the main taproot. So we haven't had a close look at the data yet, but certainly, yeah, you'll hear more from Liz in a moment.
We're working from the Wake campus in Adelaide, and it's part of Jason Brand and the team at Agriculture Victoria, and it's located.
So the first trial is looking at the impact of different grass selective herbicides on a range of different pulse crops and their impact on nodulation and nitrogen fixation. We've got Samira faba bean, Hallmark lentil, Genesis 090 chickpea and Timok vetch.
So 3 different herbicides have been applied across the 4 legume species. That's Ultro and Propizamide applied IBS at a recommended and higher rate, and then Clethodim applied at either recommended or higher rate at the 5 node stage post-emergence. We've gone in, Jason's group's gone in and they've done a whole lot of sampling for us and sent it back to the Wake campus. We've assessed both nodulation and also peak biomass for the crops so far. What we've found with nodulation is the vetch, lentil and bean were extremely well nodulated. There's a background of these rhizobia at the site. They did very well, and we didn't see any significant differences with the different herbicide treatments on the nodulation of those crops. With respect to the chickpea, that was quite poorly nodulated across all treatments despite inoculation, and there isn't a background of chickpea rhizobia down on the flat, so we needed to inoculate. Even so, we achieved poor levels of nodulation, and this is something we have seen repeatedly across the southern region with chickpea, and we're working further to understand why this poor nodulation is occurring. One of the things that might be happening with chickpea, which we did see at this site, is some quite high levels of crop root disease, which could be impacting on the nodulation process. With respect to herbicides, none of the herbicides significantly impacted on the nodulation of the chickpea. So nodulation is measured early in the season, around 10 weeks after crop emergence, and peak biomass is measured later in the season, around mid-pod fill after flowering. And from this data, we've seen that none of the herbicides have had significant impacts on peak biomass at this site. But what we're going to do now is do some further analysis so that we can look at the proportion of nitrogen that's actually come from the biological nitrogen fixation process. Root diseases and pathogens. in the new year. So there's a couple of things I'd like to draw your attention to at this point. The first In addition to that, the DNA-based test for Group EF rhizobia will be provisionally available on the Predictor B testing platform to Predictor B accredited agronomists early next year. The second thing I really want to talk about is the risk of using seed-applied chemicals when you do need to inoculate. So if you need to use a seed chemical dressing to control root or foliar diseases, and you also need to apply a rhizobial inoculant because you have a responsive site, then there's a couple of things you can do to reduce the risk. The first one is to apply the seed chemical dressing early and apply the rhizobia onto the seed, firstly at a higher rate and maybe double rate, and second as close to sowing as possible, and sow that seed into moist soil to reduce any additional stresses such as desiccation. Also avoid sowing into hostile soils such as acidic soils if seed chemical dressings are required. Further to this, you can consider using a granular inoculant with a high count of rhizobia in it, which allows separation of the rhizobia from the seed chemical dressing. So at SARDI and at the University of Adelaide, there's work going on as part of the nitrogen fixation project to look at the different seed chemicals, also chemicals applied in furrow, and different tank mixes and fertiliser mixes and their interactions with rhizobia. So keep an eye out in the future for recommendations in this space. So the final trial at this site was a chickpea trial. This was also put on the more stressful sandy soil at Nhill, and we were aiming to improve the nodulation of chickpea with a range of different strain and formulation treatments. And this was in collaboration with Jason Brand and the group at Ag Victoria. Unfortunately, this trial was hit very badly by disease, and so we haven't had many results out of it, but it is work that's happening also in South Australia, and so we hope to have some more information and do some more work in this space into next year. So to summarise, the GRDC nitrogen fixation project in the southern region is looking at a whole range of research questions to improve the nitrogen fixation and nodulation of key pulse legumes.
So at the moment we're standing in the lentil trials. So this particular trial here is the germplasm comparison in compared to what we've got up in the hill there on the sand. So same set of varieties, but just grown on a different soil type. So really interested in the relative response across those different varieties. We also have over to the other side here another repeat of the trial that's run at Cario. So just looking at sowing direction and different varieties and looking at that pod drop, pod loss trait, and just seeing how we can handle that with different row spacings and different sowing directions. So this here is the good soil patch for the chickpea germplasm trial. Up on the sandhill, we didn't even show you any footage because they really haven't grown that well. So in this trial, we deliberately chose a range of germplasm, stuff that should be adapted to the calcareous soils and the alkaline black cracking clays that we have around the Wimmera, but then also some more acid tolerant or sandy tolerant lines, earlier flowering lines to try and get that spread of germplasm. I guess what I wanted to do now is just draw your attention to this particular line here that's going to be released very soon. So this is Sisa 1352. It's a large seeded Kabuli type, so well suited to the Wimmera. We all like growing our Kabulis down in this region. So I just asked you to go to the Doan virtual tour and you'll hear a lot more about this variety and its release name and its availability for next year.